We are going live now.
Columbia University Faculty of Science Alumni Association, CUFSAA, proudly presents a distinguished speaker series to uplift the knowledge of aspiring students and budding scientists in the Faculty of Science at the University of Colombo and Sri Lanka as well. In this series of lectures, CUFSAA features accomplished scientists and key opinion leaders in different science streams to present the advancement in their field of expertise. This seminar series is a collaborative effort between CUFSAA and the Columbia University Faculty of Science. Welcome to the Distinguished Speaker Series of CUFSAA.
All right, good morning, good afternoon, good evening to everyone joining from North America and Asia here. Let's see, move on to the next slide. For some reason it's, okay. So welcome to all of you and for this speaker series in 2026. My name is Thilani Vibada. I'm an associate professor and plant breeder for international programs at Michigan State University. I'm very excited and proud to introduce one of our own graduates from Michigan State to you, who is the distinguished speaker today. And before I introduce Thilani officially, I would like to mention that how this session is going to run. There's going to be a 15-minute Q&A session at the end of the talk, and please type your questions in the Zoom chat window or the YouTube live streaming comment section. That way you don't have to wait till the end to ask your question and forget what you were going to ask. So as the talk goes on, please type up your questions. All right, so I'm just going to minimize this a little bit so you can see a brief biography of Thilani. But I'm just going to mention that that I have known Thilani for a really long time, personally as a family friend and also professionally as she was a student at Michigan State University. She is, as you can see here, she is currently a postdoctoral fellow and the breeder manager at the Department of Molecular Biology and Genetics at the Aarhus University in Denmark. And before that, when she was at Michigan State University, she actually did her bachelor's degree with a dual major in botany, plant biology, and genomics and molecular genetics from Michigan State University. And then she went on to completing her PhD degree also at MSU in the plant breeding, genetics, and biotechnology program. And she worked on potato genetics under Dr. Dave Douches, our potato breeder and geneticist at Michigan State. So, and during her graduate studies, she was selected as a plant science scholar and at the Nobel Research Institute, and she also did some internship work at J.R. Simplot Company, a potato company that we have here in the U.S. And she also has quite a bit of fellowships under her belt. I'm not gonna go into detail because it's gonna take time off her talk, but just want to mention that as she now manages the Breedome project at the institute, and she aims to automate critical steps in plant transformation and genetic engineering using robotics and artificial intelligence, and she works on legume crops right now. All right, now with that, I will give the opportunity to Thilani to go with her talk. So, let me unshare my slides so Thilani can share her slides.
Thank you for that introduction. It's also very nice to see so many familiar faces, or at least names in boxes. And so give me a few seconds to get my presentation up. Hopefully. Okay. Thumbs up, it looks good. You can hear me? Great. Okay. Hi, everyone. Thank you for inviting me to present today. Like I said, it's exciting to get a chance to share my research, and it's crazy that I am now a distinguished speaker. But today I'm going to be sharing with you some of the work from my doctorate, which, as Cho mentioned, I completed at Michigan State University. And this was all focused on the evaluation and development of genome editing and genetic engineering resources for Solanum tuberosum, also known as potatoes. So we already got kind of an overview of my background, but I think I'll go over a little bit again, just because I had such a nice academic experience at Michigan State. That's kind of set me up for the future and the career that I'm currently pursuing. And so, yeah, MSU had a lot of great opportunities in the plant sciences, and it gave me the chance to explore many different topics and sub-disciplines, even while I was working on my bachelor's. So I did things in basic plant biology, understanding genes involved in phosphate signaling. Then I also did some things with plant pathology, understanding different pathogens and their effect on some crops. And then I also worked in some more applied plant sciences, and I even got a chance to taste the early genome editing research that I pursued during my PhD work. And so all of those experiences really kind of came together to build my current research interest and the career that I'm currently pursuing and developing. And this next step here at Aarhus University in Denmark, where we're now pursuing more research in developing genome editing tools, but in legumes, as well as kind of opening up into the new frontiers of where genome editing is heading for plants, particularly in this automation of plant transformation and gene editing. And so I'll mention a little bit of that at the end, but most of today I'm going to focus on kind of my doctoral work, which was with potatoes. So, potatoes. First, let me introduce to you this crop that I have become very familiar with and will be the focus of our talk. So, potato is one of the most produced crop commodities in the world. I think most people have a potato dish that they're familiar with or enjoy. And there's about 376 million tons produced annually. And so although it has this immense economic importance, the genetic improvement of cultivated potato has lagged behind compared to other leading food crops. And this is largely because it has a really complicated genome. It has a lot of DNA that isn't really useful. It also just has a lot of DNA, it has many copies of all of its genes, and it makes everything about studying and improving the crop really challenging. And on top of that, it also has a lot of fertility issues, so it's kind of hard to make crosses and produce new generations. There's a lot of hidden issues that we unravel as we go. All of this, the conventional breeding techniques that we typically would use results in really slow progress. It often takes at least a decade to release a new potato variety. And particularly as we're seeing changes in our climate happening quite rapidly, we're really kind of thinking that we need faster and more precise tools to create targeted change. And so because of this, there's a lot of interest in developing more efficient systems to improve potato. And one option of this is to employ advanced molecular techniques like genome editing that has the opportunity to bypass these lengthy breeding cycles. Okay. So, for those of you who may not be familiar, I'll just provide like a basic introduction to genome editing in plants. So genome editing is a method that uses molecular tools to make very specific alterations to the DNA of living organisms. This can now range, the technology that we have available now, like the changes that we can make is nearly any type of genetic variation possible. But most commonly, it's used to insert, replace, or delete a specific sequence of genetic material. In plants, this can be divided into two main applications. Either it's used for basic biology as a way to characterize gene function by making these specific DNA changes and seeing how this affects the plant. Or it could be used for applied biology as a new breeding technique to make targeted improvements to enhance economically relevant traits. Although there are many tools or reagents that can serve this function, the most popular platforms that are being used today are either TALENs or I think probably the most popular one now is CRISPR-Cas9. The way that genome editing can actually work is that it uses these sequence-directed nucleases that have two main functions. First, it's to identify a specific sequence of interest that's specified by the user, by the scientist, and then it must create an alteration to that sequence. And most commonly, this is done through the catalytic activity of a nuclease, which creates a double-stranded break to the DNA. And so after this DNA break occurs, the DNA is then mended through endogenous or the native response from the plant for DNA repair. So the actual targeted alterations then occurs from exploiting this double-stranded repair mechanisms. There are two main double-stranded repair break mechanisms that occur in plants. It's either non-homologous end joining, where the broken ends are basically just kind of stuck back together or ligated. And this mechanism can be quite imprecise, and it often introduces small insertions or deletions to where the cut site occurs. There's also homology-directed repair, and this can be used to either integrate or replace a sequence when you provide the cell with a donor template. But what does this actually look like in practice? So I've broken down the process of genome editing in plants into kind of three main stages. First, we have to identify our target. Then we have to implement the actual modification. And then finally, we have to evaluate the outcome of that modification. And although this overview sounds really simple, in practicality, the actual steps can be really challenging with a lot of troubleshooting and optimization involved. So I've decided to structure this talk in a way where I'll walk you through each of these stages and describe the process. And then I'll describe how the work during my PhD dissertation attempted to address these issues and challenges, these common issues and challenges that, but within the case study of potato. So first, we have to choose a target and find its DNA sequence. The first challenge that we face is that there actually is currently a relatively small set of economically relevant breeding targets that can be improved using genome editing. Here I have a list of some of what has been implemented in potatoes so far, but it's small enough to fit on a table. This challenge of identifying targets is in part due to the limitations that I'll be describing throughout this talk that not only prevent the application of genome editing as a new breeding technique, but also prevent means of validating these economically relevant targets. So when you do actually have a target you're interested in modifying, this problem in identifying target sequences often relies heavily on having some kind of reference genome sequence available. Although now it is possible to uncover the entire sequence of an individual's genome, in many cases, there's still like a reliance on a reference genome of a single representative individual of that species or a closely related species. And the reality is that there is a lot of sequence variation between individuals, even within the same species. And to add to this complication, structural variation is also common. So by this, I mean that between two individuals of the same species, there could be many different copies of the same target sequence, or different rearrangements or different order, all complicating the identification of your target. So one solution to this challenge is to develop a model system for your species, which pairs an individual that is amenable to these advanced genetic engineering techniques with a genomic resource that allows you to more readily identify your target sequences. So this takes us to kind of one portion of my dissertation work, which was to create kind of a model system, a model organism in potato, and create a genome assembly to be used as a reference for identifying targets. And so for this, through a screen of germplasm, we came across this clone. Its name is 1S1, and it had a lot of characteristics that we really were looking for as a model organism. It's something that's highly amenable to these genetic engineering techniques. It has good fertility traits, so produce lots of viable pollen. You can make crosses and self-pollinations quite easily. It had nice tuber traits. Since we are working with the potato, we want something that looks kind of at least like a potato. And it had a relatively simple genetic background. So the DNA was not as complex as some of the other potato clones that you could work with. Briefly, I'm mentioning all this about genome assembly, so I wanted to just kind of give a very high level overview of like what this actually entails. And so we collect a sample. Usually it's kind of from leaf tissue from a plant. You grind it up and you isolate the DNA. And then from that DNA, what's in your tube is kind of a semblance of many different fragments and pieces of your original organism's genome. There's different sequencing approaches or methods that exist today. And this has been like a rapidly developing field, and the cost of sequencing has also decreased quite a bit. And different tools will have different advantages and disadvantages depending on what you have. But they all fundamentally have the ability of reading every single base or nucleotide in that DNA. And what you get as an outcome is just like a library of a bunch of different little fragments of DNA. And then you have to piece them all back together to find the longest continuous sequence. And so this relies on having overlap between the different sequences. And the hope is that you have enough overlap and enough little pieces that you can actually assemble the entire genome in the end. And this is actually something that is possible. I think at first, in the past like 10 years, like things have really adapted quite a bit. And now this is something that people can do quite easily and readily, and even in complicated genomes like that of many plants. So in the case of the genome assembly that I assembled for potato, I primarily use the sequencing technology Oxford Nanopore. The advantage of this sequencing method is that it produces like ultra long reads. So it makes it much easier to find these overlaps and stitch the pieces together. And this is particularly useful for plant genomes because they contain a lot of like small fragments that are like the same thing over and over and over and over again. And it's hard to know how many times that little repeat exists if you don't have a really long sequence that can like stretch across it. And so these are some common statistics that are used to like summarize a genome assembly. But the main thing that we look for is some kind of assessment of completeness and total size. And a common method of assessing completeness is using this BUSCO score. And so this is the benchmarking universal single copy ortholog. So it's basically just like a set of genes that we know are present in plants or some like related species. So if those genes are present and you've captured all of them in your genome assembly, then you've probably done a good job at assembling the genome. And so in the case of the genome that I was working on in my PhD, I had over 99% of these benchmarking genes present, which was a good sign that this was quite a complete and contiguous assembly. And so now all of this material, both the germplasm that I described, like this 1s1 clone, as well as the genome assembly, are publicly available. This has been summarized in a manuscript that was published to G3 Genomes and Genetics. And yeah, we wanted this to be a resource that people could use. So you can find the germplasm from the USDA Potato Gene Bank. Even the raw sequencing data is available on the NCBI's Sequence Retrieval Archive. And then you can find the genome assembly and the annotation files. So if you wanted to probe and look around at what the DNA contains, or maybe if you just want to look at like what is a genome assembly even look like, you're welcome to just go to SpudDB, and it's all free, and there's little like servers that you can kind of play around with to get familiar with what these types of files actually look like. So now let's talk about the modification steps. So this is where the process faces like the most limitations. And this is because these steps are very specifically catered to the system you're working with. This is a very like all the methods that are used in plants are very species and even like genotype dependent. And there's also a very large array of different combinations and things that you can try. So it almost seems like an unlimited list of possibilities, which can be kind of daunting, especially when you think of the scale that we do research now, which is usually just one researcher or maybe a few in a small team working together. And so it can be really tedious or challenging to troubleshoot and develop in order to find something that actually works. So the first part of this process is to choose your genome editing reagent. Earlier, I mentioned Talon's and CRISPR-Cas9 as being these very popular reagents, and they are the most popular, but there also is like a whole toolkit of new variants, particularly in this CRISPR-Cas realm, that are being either discovered from nature or being engineered. And they're specifically interested in expanding the targeting scope, so being able to target more regions without limitation, but also increasing specificity, so there isn't a concern that you're targeting any other part of the genome other than what you're interested in, as well as improving efficiency, so that you're more likely to see an editing outcome when you set up an experiment like this. So I'll take a brief moment to dive into just like CRISPR-Cas9, since this is the most popular one. The most popular kind of origin of this system comes from its native bacterial system where it was discovered, which is in Streptococcus pyogenes. So whenever you see this SP Cas9, this is referring to this bacterial gene. This variant is restricted in its targeting scope to only sequences that are adjacent to a protospacer adjacent motif, or a PAM. And the case in CRISPR-Cas9, it's always the nucleotides NGG, where N can be any nucleotide, but you need two guanines next to each other. And then what is upstream of that can be a sequence that you can target. So if you, this can be a limitation, because if you're trying to make a very tailored alteration and there is no PAM site in the region that you're interested in, you would not be able to use this system. So like I said, this is why people are now trying to study and alter the engineering scope of these enzymes. Okay, and then next, after choosing the reagent, you now need to find a way to deliver them into your plant cells. There's many different techniques. This is only like a brief summary of them. There's actually a lot more than just this. But the gold standard is using agrobacterium. It's a species, or it's a strain of bacteria that already had this native mechanism of inserting its own DNA into the plant. It did this because it wanted the plant to make a nice home for it. But now we, as scientists, genetic engineers, we've exploited this mechanism so that we use it to insert the DNA that we like into the plant instead. And this is definitely the most well-described transformation method. But this also runs into many challenges, because you'll find different susceptibility of different strains of agrobacterium compared to your different plant species, and you have to try all of these different combinations to see which one is actually going to work, if it even does work, because sometimes it doesn't. And then if it doesn't, people tend to then move into these other approaches that are a little more experimental or expensive. And so with every step, there is kind of a trade-off in your choice. There's kind of also two overarching ways of delivering genome editing reagents. It can either be through a stable transformation, and this is, like I had just kind of described with agrobacterium, this is like where the DNA that you're interested in is actually incorporated into the plant's genome. So this would be all of those genome editing reagents, the CRISPR-Cas9, the target sequence. All of this stuff is put into the plant genome. It's expressed by the plant, and then the targeting happens in the cells following that. You can also do transient transformations, and this is without having to put anything inside of the plant's genome. And this is often using ribonucleic proteins, or RNPs. And this is basically where the active form of the CRISPR-Cas system, as a protein and its RNA, is directly put into the plant cells. And so this is a means that you can kind of bypass some regulatory concerns in some countries where this transgenic stable transformation techniques are not favorable. After you've settled on what approach might work to introduce the genome editing reagents into the plant cells, you'll have to find a way of recovering those independently transformed cells as a whole plant. And this is definitely the most troublesome part for many plant species, because it requires a highly time-consuming tissue culture phase, where these transformed plant cells are exposed to different hormones that dictate their development. And together, this process is referred to as regeneration. I have some examples below of some pictures of regenerating explants and potato. And what you'll often see is that these are both potatoes, two different genotypes, but on the same exact media, and they'll respond in wildly different ways. And so this is what I mean about like genotype dependence. So this one is regenerating really, really well. So the outcome that we want to see is like new shoots being formed. I know this kind of looks crazy. It is actually pretty crazy, but this is like a shoot of a plant that you can then put into a little box and it'll form roots. And then this is just dead leaves because nothing ever happened when it was grown on this media. And so this is the type of troubleshooting that we often face is you have this endless amount of different plant species that you could work with and also different genotypes of that plant species, and you don't really know what is going to work until you try it. And in addition to that, these methods are also quite time consuming. And so at least in this approach that I've shown at the bottom, you can start to see the signs of shoots developing after about 1 month. But the whole process takes about 3 to 6 months. But there are some species where you'll have to wait even as long as 1 year before you can collect, in some cases even longer, before you can collect a regenerated a plant. And I know that timeline doesn't fit so well into a graduate student study. So these are the types of things that make this work kind of quite challenging. And so timing and being able to make decisions quickly and knowing when to move on is also very important in the line of work. So as you can see, the steps of actually modifying a plant using genome editing can be quite challenging, particularly as we attempt to step outside of like our model systems. And in order to really employ these tools, it can take a lot of time and effort to test, to evaluate, and optimize different methods. And so one solution to this is to identify germplasm that is already amenable to the methods that have already worked in your species. If you happen to be someone like me who works in a crop where people have already began to work on a species. In the case where you're working with a new species that no one has ever worked with, which is the challenge I'm facing now, working in the different legumes that I work with in my postdoctoral work, is you first kind of start with a related species, maybe some other model legume, but it is a larger jump and it makes it kind of hard. But yes, so in potato, we had the benefit of potato being quite amenable to genetic engineering, or there were some lines where there are approaches that worked. So we leveraged that and just tried to find specific individuals that could work with the methods that were already available. So for this, this was something that I did kind of early in my PhD, which was just to screen as much potato germplasm as we had available that had really amenable and nice traits, at least for our breeding program. So first I was looking at tuber quality traits, so things that were kind of nicely shaped and fit within our breeding goals. Then I was looking for things that had good fertility traits because we want to work with things that we can continue to work with past the initial step of doing the genetic engineering. But then we also needed to actually be amenable to these regeneration and transformation and genetic engineering techniques. And so here's another example of kind of the wide array of responses when you put different genotypes onto the same media. Another thing that varies, it's not just the genotype themselves, but it's also the tissue you choose. So in theory, you can actually take any part of a plant and attempt to regenerate it because all plant cells, they're almost kind of like a stem cell, but like it's slightly different, but they all have this capacity to be redirected in their development. And so it's not determinate like our most of the cells in the human body. So that is something that we can leverage. And so in this case, I was trying internode and leaf tissue. So internode is kind of like, it's basically like the stem of the plant, as well as leaves. And so depending on the genotype, but also depending on the tissue that you start with, you can see a wide variation in the response to this hormone media. The two that in this study that performed really well was 1S1, and this was the one that we did the genome assembly for, as well as this clone 73705, which I liked a lot, but did not make a genome assembly for it. People actually are using this for genetic engineering studies as well. Okay, so the main takeaway here is that employing methods of genome editing in plants requires strategic planning. You really have to consider each step of the process carefully and try to consider your best options based on previous literature and experience. Because of how time-consuming these methods are, it's also common to pursue multiple options at once. These methods are also often kind of described as more of an art or a craft than it is a science at times. Maybe it's more witchcraft. I don't know. I've heard it described many different ways. So I think it's important that you also take a lot of pictures and provide more like descriptive notes. It's not just data and numbers. It is a very visual type of science. And it's also really important to share your experiences. Most of what we do in this kind of work doesn't get published because it's just stuff that doesn't work, or it's just a piece of an incomplete puzzle. But sharing your experiences with other people in the field is really important so that we understand what people have already tried, and maybe there is a piece that someone has already accomplished that could help you through the process. So communicating and participating in different workshops and speaking with the strangers is also a part of this job. And so, yeah, it's quite common to get negative results, but it's really that incremental progress that's important in this line of work. Okay, and now it's the final stage where you finally evaluate the outcome of the modification. So you'll have to then screen for edits, for the edits that you made at your target site. There's many different methods that exist, and they'll range in cost and sensitivity, with more sensitive methods requiring sometimes more time and costing more. So depending on the scale of your experiment, how many individuals that you end up having that look transformed, you'll choose a different approach accordingly. But all methods of screening first rely on amplifying your target sequence through a PCR. So this is where you take your DNA and you can amplify it so that you have another little copy of it, because we already know so much about this template. In the least sensitive methods that are seen at the bottom of this table here, they can only tell whether there is an edit, in the sense that it knows that the sequence is different from what it should be, but it can't tell you exactly what happened, like what changed in the sequence. And then as you get to some of the more sensitive methods, like this amplicon sequencing or cloning that's at the top, you get a very clear picture of exactly what occurred into the DNA, like to the exact nucleotide, the exact base, what occurred. Okay, and so here, a notable challenge, one of the notable challenges when it comes to screening edits in plants is it's actually really challenging to get what we call heritable editing. It's more common, or it is quite common to see what we call chimeric cells with mosaic edits. So it's like you have a developing plant, and then each cell of the plant has a different edit inside of it. And some of them are maybe not even edited at all. Some of them retain the wild type sequence. But then when you try and pass that individual on to the next generation, you can totally lose those edits that occur. And so that's what we mean by heritable editing. We want the same edit to occur in all of the cells of the plant so that we know that it's going to be maintained and propagated as the plant grows and develops. And this is an example of of kind of what that looks like. This is like a bunch of different like sequencing reads, and like each line is a different like sequencing read. And then I just highlighted the variation, or the points where there's something different. And so in this individual, if it were truly edited at most, if it was truly a heritable edit, at most we would only see 2 different types of edits. But here we see quite a few different variations. And in order to decipher this, it requires quite like a sensitive screening approach. So something like those sequencing methods that I said at like the top of that table. Because if you use one of these insensitive methods, all you know is that an edit occurred, and you don't know exactly what is contained in the cell itself. So this is all kind of describing how we would screen for your on-target edit, so like the thing that you know that you want to make a change to. But there's also this concern about off-target editing. So what happens in the rest of the genome? I mean, you're targeting like a piece that is maybe like a 20 bases of something that is like gigabases in size. So what happens to the rest of the DNA? And so off-targeting editing is something that people are concerned about, especially if plant genes tend to have many copies, or copies that are very similar to each other. So especially if in the sense of getting crops, food crops out to the fields, having a screening approach that encompasses the entire genome, so you know what occurred even outside of your gene, is very important. But fortunately, like I said, we actually do have these techniques available to us through these whole genome sequencing tools. It just comes with a little bit more of a cost, so it still requires some time and effort to get to that place. But off-targeting, it can occur, and some Cas variants, or these CRISPR systems, they can tolerate some mismatches. So maybe 1 or 2 nucleotides in the target sequence, if it varies in something outside, there is the potential of off-targeting. And this will also vary depending on the reagent that you're using. It has been shown, though, quite reliably now with CRISPR-Cas9, that if you design a guide with good if you design a good guide, you will not see off-targeting. And a good guide is something that like doesn't have any potential mismatches within particularly the first 7 nucleotides of the guide, adjacent to this protospacer adjacent motif. It can, if there's mismatches outside of that, it often doesn't recognize that sequence. So there's actually been quite a lot of studies now that have kind of shown the same trend. But this is something that we'll have to continue to evaluate as we get these new reagent systems developing. Something else that we have to be concerned about at the end of this whole process is something called somaclonal variation. And this is caused by this process of tissue culture regeneration. So in tissue culture regeneration, we're exposing those plants to really high levels of different hormones. And you can almost kind of think of them as like potentially like a carcinogen. Like they can be mutagens. They can make adverse changes to the plant as well. And so even just going through that tissue culture process, what you started with that looked like, a nice plant, it might come out looking totally different. And so this is also something that you have to be aware of when you develop these methods. And so usually one of the first steps before you even get into gene editing and transformation is just seeing, can you get through this tissue culture process without, while maintaining the identity of that individual. There's also a lot of development in the field of doing tissue culture free approaches. But right now it's kind of depending on your species, there's different genetic hurdles, but also it's a question of scale. It's often quite inefficient, and you'll need larger screening programs in order for those types of techniques to work. And then finally, there's also the effect of pleiotropy. And so even when you are successful at making your targeted change to that gene or sequence that you were interested in, that gene may also be involved in other functions that you hadn't anticipated. And so this is always the risk that we make when we make alterations to plants. And this is even true in conventional breeding approaches, is that most plants, basically all genes, they don't just like stand alone. They're involved in complicated networks with other genes and within a larger system. And so you make one change, it's going to result in some other changes to the plant. But the main thing that, at least in the plant breeding sphere, that we're concerned about is that it doesn't change like yield or the phenotype and vigor of the plant. So as long as it appears the same or better, then it is kind of withheld to the correct standard. And like I said, so like the solution to this approach is leveraging these like generating sequencing, whole genome sequencing methods, where we can then screen the entire genome of the plant and then actually identify the targets and off targets as well. And then this is just my final chapter of my thesis. I know I think I'm maybe running a little low on time, so I'll go through this, but stop me if I'm taking too long. So when it comes to this off-target screening, I did a project where we did a genome-wide evaluation of off-target editing using CRISPR-Cas9 in seed and vegetatively propagated crop species. This project was a part of a U.S. Department of Agriculture Biotechnology Risk Assessment grant. And so I'll briefly go over the design of the experiment, and then we will use it to understand how next generation sequencing can be used to evaluate the outcomes of a genome editing experiment. Since we've already been talking about potato all day, I'm only going to focus on the work in this project that was related to potato, but some of the same experiments and evaluations were done to the seed-propagated crop species Camelina sativa oilseed. It's an oilseed crop. So first, we're going to identify our trait. And in this case, this was the tuber-expressed polyphenol oxidases. These are a really well-characterized trait and enzymes in different plants. And it's basically the thing that controls, like if you cut a potato or if you cut an apple, when it begins to brown, this is due to this release of an oxidative species, and the expression of these polyphenol oxidases kind of control this phenotype. But this can be a large contributor to food waste. When fresh produce is cut, if it begins to brown too early, it'll get thrown away. And so this was something that had economic relevance to the potato industry. So we were interested in using this and it has also been well studied in potatoes as well as other species. So it was a safe target to use for this experiment. We then identified the PPOs, or polyphenol oxidases, that were expressed in tubers, or like in the potato itself, since that was the tissue that we were most interested in. And then we designed a single guide that could target all of the different copies simultaneously. The CRISPR-Cas9 genome editing reagents were delivered into the plant cells using agrobacterium-mediated transformation. So this is like that stable transformation method where it uses bacteria to put the DNA into the plant, and then the editing happens afterwards. And then the plants were recovered through regeneration, like the photos that I showed you with the plates, with the leaves and the shoots coming out. In this experiment, it resulted in 27 stable transformants, so 27 independent little like shoots that became whole new plants. But out of those 27 that actually contain that piece of DNA that we put into it, only 7 of them actually had detectable edits. And this is quite a common outcome when it comes to plant transformation. Plant gene editing is like that challenge I mentioned, the challenge in getting heritable editing. You can get the editing machinery into the plant cells, but the editing occurrence in your target is like an independent event. So it's another step of the process that needs to be optimized. Then in terms of screening or evaluating and screening the on target, I first employed a less sensitive approach, just so that I can screen all of my material at once, and then identify the ones that actually did have an edit, like those 7. So I screened all 27, and then I focused in on the 7 that had the edits to do a more sensitive approach, like this amplicon next generation sequencing. So on the top is an example of the output from using this online tool called Synthago ICE. And it assists in identifying insertion and deletion mutations from the chromatograms of like Sanger sequencing. So this is one sequencing approach that is generally, it's quite cheap and people use it quite often. But it'll contain like a mixture of the different copies of your target. And so then I also employed next generation amplicon sequencing to then resolve the ambiguities between the 2 different copies at this target site. And so an example that's shown here is that in the wild type, or in the reference sequence, there is not an A in this position, but in the edited sequence, there is now an A. And then in the other allele, or the other copy of this gene, no change was made because the reference and the target, or the reference and the edited individual maintained the same identical sequence. And so across the 7 edited events, no event had like a complete knockout where all of the genes and all of the alleles were edited and targeted. And in basically all cases, only one allele of one target sequence was actually edited. And this highlights a common challenge in the poor editing efficiencies that when you work in non-model systems. The types of edits that did occur ranged in size from a 17 base pair deletion to a 1-to-1 base pair insertions. And there was one outlier in one of the events where it had this mosaic editing, where I could see many, many different types of mutations coming up from like the sequencing reads. And then following the validation of the on-target editing, we produced whole genome sequencing for all of these edited events in order to evaluate the editing outcomes at like a genome-wide scale. For this analysis, I used the tool called Cas-Off Finder. I used this to annotate all of the putative, or all of the potential off-target sites in the genome assembly. This indicated that there could be 898 potential off-target sites. So I was looking for things that were similar enough to our target with only like 5 or bases different. And so that does bring up a lot of different options with 898. Sounds a little scary. But out of all of those sites, only 2 events from the sequencing showed that they had overlapping variants in these regions. And then I went and I really just manually inspected this alignment, and I could see that the variation that was being pulled out in this region was actually shared with the wild type kind of control plants. And this is also kind of common when you have like a genome assembly. It's also natural over time. You create a genome assembly from an individual, but it's natural over time as you maintain a plant that it'll start to accumulate new mutations, things that you don't really see. And so that's why it's important to also have these wild type controls, so that you can see that there is natural variation in the plant that is not related to the editing outcome actually that we were doing. So this was not an off-target. This is not related to the off-target editing. This was just a variant that arose naturally from the plant prior. And so this actually overall indicated that there was really negligible evidence of CRISPR-Cas9 dependent off-target editing. And so we show this in our study, but this has also been seen in several other crop species now, that with a well-designed guide, CRISPR-Cas9 is actually quite specific. And so for the conclusion of this section, in terms of off-target editing, CRISPR-Cas9, it's already been reported as being quite specific, and also across many different species. And the majority of this variation that we end up seeing in plants is more likely to be originated from spontaneous variation that just occurs from the natural propagation of plants than it is due to editing. And overall, incorporating next generation sequencing provides a comprehensive view of the editing outcomes for both the on and off target editing. So today I walked you through the process of what it looks like to do genome editing in plants. I hope you can see that this process is not so simple, but with concentrated efforts, improvements can be made. We looked at several of the most notable challenges and made attempts to try to remedy them in potato, although there is still a lot more to explore. I think definitely my hope when I was working on my PhD was just to try some things, explore new topics, and at least make an incremental contribution to the field. And so I was able to improve a few of these steps, as well as provide some resources for future studies. But now we're at this new point of where is this field headed, and what is the direction that we're going toward. And so, like I had kind of alluded in the beginning of this talk, the future direction that we're seeing in the field of plant genetic engineering is heading toward leveraging robotics to automate these steps, because this is everything that I described to you is very time consuming. It takes a lot of effort, it takes a lot of personnel. There's a lot of reasons and ways it can go wrong. And given how quickly artificial intelligence and robotics has expanded in only in the past few years, it is definitely something that a lot of fields are trying to leverage to make these challenging, cumbersome manual steps much easier. And so now I manage a program. I coordinate many universities in Denmark. I've created a network where each university is kind of focused on automating a different critical step in this process. So basically, every step that I described to you today, from selecting and assembling the reagent to developing the plant transformation pipeline to screening all of these events and doing the transformations to actually automating the sequencing detection. We now are trying to leverage automation and robotics to automate each of those steps. So each university is focused on a different aspect of that, and I'm kind of coordinating the efforts so that we can actually make it happen. And so this is a very new project. Our kickoff was just in April, So this is a lot of ideas, and we're in the phase of like purchasing and designing equipment. And so this is funded as an 8-year project, but we're establishing core facilities so that they can be maintained long term and eventually be a client facing resource where we can potentially be producing different methods or transformation events or reagents for people who would like it and can pay for it. And so, yes, thank you. I have so many people to thank, but most of what I described today was from my PhD work. So, of course, thanks to my PhD group, who I still keep in close contact with, and everyone at Michigan State. It's a lovely place to be. And so I developed a very nice community there that I'm very grateful for. And so, yes, I think that's it. And I'm sorry, I definitely went over on time, but thank you for listening to me. And I'm happy to take any of your questions.
Great. Thank you, Thilani. This is a fantastic talk. I think I learned more than what I learned from at your defense. So now it's time for Q&A. I wonder if I should. No, I don't think I need to share my screen yet. Let's get some questions and answers from others. And let's see, do we have any in the Google Doc? Okay, so I'll start with someone else's question than mine. One of the questions is, how do you decide which mutations insertions to incorporate? Can a CRISPR-based random mutagenesis library be more high throughput to find more economically beneficial variants?
Yeah, that's a good question. So there are there's two ways of approaching gene editing using CRISPR-based systems. Most often, we're relying on the endogenous like repair mechanism of the plant, where it will create these insertions and deletions. But this is at random, so we cannot actually control what the insertion was, specifically what base is being inserted or what's being deleted and at what size. There are trends to this. Generally, CRISPR-Cas9 cuts 3 nucleotides upstream of the PAM site. And depending on how you design the guide, if it is reduced in microhomologies, so like there's like little sequences that flank each other, it'll only produce a 1 to 3 nucleotide insertion. And if you put little flanking regions that are similar to each other, you're more likely to get larger deletions. But in that case, it's totally random and you can't really control, other than what we see as trends, you can't control the outcome. So it's more a question of scale and understanding that there are some trends in the type of outcome that can occur. If you do want to make a very specific alteration where you want to insert a specific one nucleotide or more or less or something like that, then you have to rely on these either the homology directed approaches or there's something called prime editing, which it kind of is almost like it provides a very short template to be inserted at the cut site. And this works if you're looking for something very small. If you want to insert an entire gene, you have to use homology directed repair. And currently in plants, this is quite inefficient. And so, yes, there are different approaches. So if you are interested in making a very specific alteration, and if it's only a few, like less than like 20 nucleotides, I think it is in size, maybe it's a little more, maybe it's like 36 or something, then you can use prime editing and you can make a very specific change. But it is also true that people are using, you can use this random insertion of different sized variants to then see if there is a variation in the outcome and change. And so it's kind of a way of introducing quantitative, a change in a trait quantitatively. And so, yeah, leveraging a wide variety of variants and then seeing if there's a variation in the outcome as well. And then that question is definitely something about scale. So I think that's...
Sounds good. Yeah, so I had a quick question. Like, how do you talked about the mosaic edit showing up on the same plant. So what do you do when that happens?
Yeah, so um, there's different routes to this. So in potato, I'll start with I'll just focus on potato because it'll be a different story with everything. But with potato, what you can do is you have this mosaic plant that has a bunch of different edits. What you can do is then just use that mosaic plant and then regenerate another plant from it. Like
Mm,
if you then it has a mixture of different cells, but when
mm,
you regenerate a plant, typically it's starting from a single cell
yep,
again anyway, and then it
yep.
kind of like purifies
Mm-hmm.
the amount of variation. Um, if it's also throughout the entire plant and there isn't a high proportion of like wild type or unedited tissue, and you work in a plant that can produce seeds, then you can try and get it to the next generation. And then through like self pollination, hopefully that can also be a way of fixing the edit. But in potato, it's not a seed propagated plant. So we tend to avoid that since we often don't take the plant to this seed
Mm,
step.
mm-hmm. Great. Are there any other questions, perhaps career oriented? She did her research work, degrees in the U.S., and now she's in Europe. Um, are there any other questions? Maybe students? All right, let me ask this until we wait for another question. So, what is it like to do your degrees in the US and now be in Europe? Because Europe is, well, maybe things are changing now. It was a little bit more anti genetic engineering, gene editing, perhaps. So, how was that transformation for you? Are things changing? Clearly, looks like things are changing, but what is the regulatory aspects looking like in Europe?
Yeah. I mean, I think there's definitely a consensus, at least among scientists, that they hold the same view that we do, like in the US, that like these new breeding techniques aren't regulatory concern as long as they're kind of maintained in this informed way.
Mm-hmm.
But of course, there has been this difference in public opinion. But actually, like the European Union is just recently they're on path, very likely to approve gene editing as a new breeding technique, and that was actually a part of this Breed Dome project. Is that we're kind of leveraging this is very good timing for us, I think, and also the Danish government is very supportive of um, these breeding tools. And I think it's particularly because um, people are aware of and they're feeling the effects of climate change
Mm-hmm.
and can see the importance of new and specialized techniques for improving crops.
Yeah, fantastic. Um, any other questions? Tilani, perhaps you can unshare and I can share my screen for while we wait for other questions, I suppose. Okay, thank you. All right, so this was our question and answer session. Can I ask if there are any other questions? Anybody, you can raise your hand if you just don't want to type anything. Okay. All right, great. In that case, we'll move on to the next slide. So we're looking for any suggested suggestions and comments. So CUFSA Distinguished Speaker Series is organized by the Speaker Series Subcommittee of CUFSA North America in association with the Faculty of Science, University of Colombo. So if you would like to contact the subcommittee, you can write to us at speakerseries@cufsa.org. We are particularly interested in hearing from you with topics and speaker suggestions for future. And then we also want to thank Professor Darshini Bandupriya, the head of the Department of Plant Sciences at University of Colombo. Thank you so much for your support. And this speaker series is organized and is co-organized with the Botanical Society of the University of Colombo, BOTSOC, which I was a part of too. So I'm really excited that this is continuing. And so I'd like to now, you know, give the opportunity to BOTSOC for final remarks. should I stop sharing or?
You can keep it on.
Oh, okay. Is it?
Hello, I'm Samadhi from the Faculty of Science, University of Colombo, Plant Science Department. I have currently finished my 4 years, so it was really nice listening to Miss Thilani's speech and presentation. On behalf of BOTSOC and the Plant Science Department of the Faculty of Science, University of Colombo, we'd immensely thank Miss Thilani for giving out her insights and telling us her experience on her PhD and her bachelor's as well. Uh, and as we are also stepping into the next stage of our future, so some of us are planning to do academic in the future. So it was really nice seeing how the things we learned in theory are actually practiced in research in postdoctoral level as well. So thank you very much, Ms. Tilani, and thank you, CUFSAA, for giving us this opportunity.
Great. Thank you, Samadhi. Appreciate your comments. So with that, thank you everyone, and we will see you all in the next speaker series talk. Thank you. All right.
Thanks a lot.
And everyone, yeah.
All right. I will. Chandrasiri, would you be ending this on your end or?